our transcription factor 3A was discovered, purified from Xenopus, and the way in which it was characterized. For example, you got this transcription factor which binds to your internal control region of 5S gene. In first lecture, we have seen that. Uh, then subsequently, we saw that this transcription factor, which binds the internal control region of 5S DNA, this same protein is able to bind your 5S RNA. That means the 5S RNA is synthesized and it has some kind of feedback regulation because the same protein, when there is more RNA, then the protein is binding to the RNA, but in the limited quantity, it elicits your transcription factor. That means it elicits, binds to the DNA and allows the transcription to occur. See, there is a basic difference between your RNA polymerase together with transcription factor and without your transcription factor. That means if you have just in vitro experiment where you have just RNA, or for example, if you are carrying any in vitro experiment, for example, this is your tube and inside that, imagine you have 5S DNA, right? So this 5S DNA, only in the presence of RNA polymerase, is not very direct. That means it can give rise to any type of RNA material, and that can happen from anywhere in the promoter this sequence. That means RNA polymerase by itself is not very straightforward. It can synthesize RNA material, but in the presence of transcription factor, for example, if you also incubate this material here with the RNA polymerase, then the transcription is very specific. That means it only binds to the promoter region and specifically allows RNA polymerase to synthesize from the +1 region, what we call as a specific RNA synthesis. So this is the basic difference between your RNA polymerase alone and together with transcription factor. So this we saw in the purification material where in the presence of RNA polymerase alone, there was a lot of background, but in the presence of purified transcription factor, the synthesis of 5S RNA was very specific without any background. So that we have seen, and after that, major breakthrough came from Miller experiment and subsequently from crystal structure. So with Miller experiment, we saw that he obtained a repetitive sequences, and this repeats had very conserved amino acids and very conserved bands of proteolytic cleavage after especially after chymotrypsin. So this we have seen, right? So after that, crystal structure actually gave us that details of amino acids specifically from -1, 3, and 6 that established contacts with 4 base pair in the DNA. Now thus, this zinc finger motifs provide an excellent framework for designing your DNA binding proteins with a variety of new sequence specificity. And in my last lecture, I said that in theory, one can design a zinc finger domains for each of the 64 triplet codon shown here, and using a combination of these fingers, one could design a protein for sequence specific recognition of any of the segment of the DNA. So this was the question, and I was asked in last lecture that how this specificity occur because there is a very prominent amino acid at this position. So this is one example here. So what you see here are the 3 repeats. For example, F1, F2, and F3. F1 and F3 are similar in all the cases, whereas if you see F2, the middle domain has XXX here. These X were replaced by different kinds of middle domain. For example, in first case, so here in the first case, it was replaced, all the X was replaced by zinc finger 268. And especially at the sixth position you have histidine. Sorry, this is seventh position. So you have threonine in this case. Similarly, in other case, you have in second case you have F2 glycine. Why? Because at the sixth position you see you have the presence of glycine. So similarly at third position, third mutant you have valine. Because of this valine presence, asparagine and arginine. So what they did in this essay, they actually kept F1 and F3 as it is, whereas in the middle domain, especially at the X position, they changed this with the different varying amino acid. And with this amino acid at the background, they had also changed the aspartic acid presence here, and they mutated it to alanine. So for example, then what you have here in wild type, you have this protein where this alanine is not mutated, whereas in mutant you have this mutation at the background, and on top of it the alpha helical region is varying at different position, especially at sixth position. And now by doing so, they express this kind of protein via making a phage. So you can imagine you have a phage display where you are expressing this protein one by one, and then in the some kind of method, you are also incubating different kind of oligonucleotides. So for example, now coming to the oligonucleotides, they had a strategy. For example, the first type they did, either they kept G, A, T, C at the first position, and then second and third position any nucleotide, for example N N. So any nucleotide was present in this case. So similarly in the second one, they had at the second position, they were very specific at placing these bases, and on the first and third position there was any of the nucleotides. And similarly same thing they did for the third position, and so this kind of scenario. So by doing this kind of phage display and selecting different oligonucleotides, one can actually figure out that which amino acid residues coming out of your helical region, and at which position you have proper selection of or specificity for that particular base. For example, this is a result from the wild type of zinc finger 268 that recognizes G and T at the first position. As you can see here, either G or T at the prominent position. At the second position and third position, it recognizes G and G. Whereas if the mutation happens, then you have, this N. N means any of these at the first position. It can also recognize G, it can also recognize A, it can also recognize T and C. Similarly, this was done for all the mutants, and you can see that there one can actually change the sequences or the specificity by changing your amino acid at the place of sixth position. And similarly, wild type recognizes GT or TC, and here it recognizes N at the first position, similarly here. So via this method, I would request you to actually mark these differences, G or T or T or C, or any of these differences where this kind of scenario is able to select either this G or T or any of the N position. For example, in this case, you have GATC. So this was the method they used for selecting specific oligonucleotides with respect to your protein DNA binding concentration. So that means the specificity of DNA protein interaction in this case can be adjusted. So how it can be adjusted? Because the motifs by within the alpha helical can be changed, and by changing this residue, one can change the binding properties of this individual fingers. So that is how one can actually play with your specificity in DNA binding interactions. So that is one thing. Apart from that, there is one more interesting thing, not only the specificity can be changed by changing the residues, but the strength of interaction and the binding specificity by zinc finger domains can also be changed by readily incorporating number of repeats. So there are two things here. First, the individual zinc finger domains, and then the number of repeats or domains. So here, if you add 2, then you have specificity for 6 base pair. But if you add more, then it will go on increasing. For example, 3 will give you 9, the 4 will give you 12, and similarly, and so on. So that means you can change the strength as well as specificity by incorporating your repeats and the number of repeats, or by changing the alpha helical amino acid position. And now this is very unique for zinc finger motifs, mainly because these kind of repeats are not accessible or not found in other DNA binding proteins. For example, helix 10 helix motifs, or helix loop helix motif, or any other motifs which binds to the DNA. So these types of repeats are not available with those kind of DNA binding protein. That's why it is unique, and within a short span of time, like for example, if you want to reduce the number or change the strength, it can be actually changed by either playing with these two aspects, right? So that is one aspect. So this we have seen in last lecture, and in today's lecture I actually did some recapitulation of last one, and then we will move further. So in today's lecture, we will basically focus on our chimeric restriction endonucleases, and specifically for zinc finger motifs. So if you remember in our last lecture, especially lecture number 8, we talked about a chimeric enzyme, which was formed basically by linking two domains, specifically one by ultrathorax bithorax gene, and by the your zinc finger nucleases. Now, similarly, by doing so, one is able to target 7 to 9 base pair. So if you see here, so it was initially found as a 7 base pair, and then it was changed to 9 base pair sequence. That means by linking UBX to FOC nucleases, you are targeting a sequence, specifically this sequence, where 9 base pair sequences can be recognized by your specific amino acids here, your UBX gene. Now, with the same strategy, they were able to now focus on zinc finger nucleases, and specifically here also they used three zinc finger nucleases. I have, three zinc fingers repeats. I have shown only one. So with these three repeats, they were able to construct one chimeric enzyme by using your 25 kilodalton 196 amino acid, your C-terminal domain of FOC1. So what they show here in this paper, hybrid restriction enzyme, zinc finger fusions to FOC1 cleavage domain. And this was published in 1996. And I will come to it why I'm saying this 1996, because by this time, Dimer's publication was not published. So dimer, because FOC1 requires dimer, was not known by this time. So he used this strategy where Chandrasekharan group used zinc finger domain, attached your FOC nucleases within your PET15B vector, and the cloning strategy is shown here, where they have his tag at the N terminus, then you have zinc finger domain here, and then in between you have linker of 15 amino acids, then you have placement of 196 amino acid of nucleases. So this was their strategy. They did the cloning, and after that they were able to purify this protein in good amount. For example, here what you see is the crude extract, then his bound column, you have Safarez column, and finally from the gel filtration they were able to purify this protein. And I, as tradition, once you have the protein or chimeric protein, then you want to see whether it is cutting any DNA or specific at the specific site. So for that they used one substrate, and here they used lambda fast DNA substrate with 48.5 kb length, and this is shown here. So specifically I should mention here, they when they were constructing this cloning, they did two kind of constructs. One was called as QDR, and another was called as a QNR. So basically, these two things were shown here. For you can say this is QDR and this is QNR sequence, and both were able to actually cut the DNA at specific recognition sequences. For example, you have more than one cut in this kind of QDR regions here, whereas in this case QNR you have very specific recognition site. That means you have one recognition site. So when you use QDR nucleases, this is able to cut at around 5.5 kb predominantly and around 43 kb, whereas in QNR it is able to cut at two prominent position, one at 9.5 kb and another at 39 kb. So so this they also had the prominent recognition sequences, for example G or G or GCT, something like that, GCG. So for this also GGA, then GAT, and then GGG. So these were the specific sequences of 9 base pair. So imagine in first chimeric, they were able to target specifically to 9 base pair. And here also they are able to target with zinc finger nucleases to 9 base pair. So this was unique, and they were able to show that their zinc finger nucleases were able to cut the DNA at a specific recognition site. Now, one important observation from them, actually they found that both of this enzyme, whenever they used it, they required a very good amount. That means a high amount of this nucleases were required for cutting these sites, even a smaller sites. So they were curious, and only after in 1998 this chimeric dimer uh, dimer sequences were known, and then they realized that why they required very high quantity of your zinc finger nucleases, or in fact UBX nucleases. So they figured out that since two domains of your FOC1 enzymes are required to cleave double-stranded DNA, then their strategy actually started changing, and then they wanted to construct a novel nucleases. So far they were able to construct this kind of motifs. For example, one zinc finger, second, third attached to the linker, and then this fork endonucleases were able to cut at specific sites, and all the specific binding sites they were able to recognize with the help of footprinting assays, where they do maximum Gilbert kind of sequencing, and they were able to figure out at what specific positions they were able to cut the double stranded DNA. So now changing, because they know that dimer is required. So in that sense, they were able to first do that what kind of orientation is required. For example, whether head to head or tail to tail kind of orientation required. So Chandrasekhar, along with her colleague Carol, actually they figured out that tail to tail orientation is required for the dimer position, or is the best for cutting the two unpaired region. And second, they wanted to check whether two paired non-identical recognition site non-identical paired sequences can be recognized by the zinc finger nucleases. And for that, they actually did this kind of strategy. So for example, you have three zinc finger domains attached to the FOC1 enzyme, and then this is recognition one separate recognition site, whereas this is separate domains or tandem repeats which recognizes different recognition sequences attached to a FOC1 enzyme. And in between they had this gap of 1, 2, 3, 4, 5, and 6. These were called as spacers. So usually they required around 6 base pair spacer region to separate these two halves. And now these hybrid nucleases were utilized in many labs. Now basically what they did here, they used zinc finger nucleases that was introduced as a pair of tandem binding sites and engineered in opposite direction or opposite orientation so that it can cut exactly at the position, giving rise to 4 base pair overhangs. So this is the actual, at last they were able to figure out. So before this they did lot of experiments and lot of experiments where they wanted to find out what orientation is best, then what number of spaces is the best. And I will give you just a preview, like just an example that how many experiments they would have done. So this is one example there where they see that what is the lower limit of spacer. So this is 4 base pair. And what is the higher limit of your spacer? So this is a 40 base pair. So that means when they go down up to 4 base pair spacers, there is no cut and it cuts around at the same position or different from 6 base pairs to onwards to 35 base pairs, whereas 40 was the higher limit. So based on these kind of experiment, they were able to figure out 6 base pair spacers is working best with the opposite orientation in this case. And similarly, they also figured out what are the orientation for these two tandem repeats, exactly this half or the other half. So now this kind of enzymes were used by Chandrasekharan as well as the Dana Corral, who was working on Drosophila. So they therefore she worked on this and she was able to prepare some kind of mutants with using this kind zinc finger nucleases. And later this Dana Carroll together with another scientist, namely Porteous, he was also using this zinc finger nucleases where he used GFP and changing GFP specificity in HeLa or human cells actually. So these kind of application, they were already testing it, and later these two person were responsible in actually creating a biotech company named Sangamo. So this was the total history of your zinc finger nucleases, and the prominent hybrid enzyme which is used by most of the labs is something like this. Okay. So as I said that there are advantages that this is modular and can be used by different nature, but along with that there are few limitations and we will see what are the major limitations of zinc finger nucleases. So there are majorly two limitations. One is zinc finger domains have limited modularity. As one end I'm saying this is modular, but another hand I am also saying this is uh, limited modularity. That means this actually has a context dependent DNA binding effects. That means if you remember the experiment which I showed you, so at position number 2 if you replace from D to A, so by replacing this kind of mutation at position number 2, this preclude of having some kind of binding site either to G or either to C, something like this. So that means you cannot actually create one independent finger and second independent finger. So if you have certain amino acid at some helical region, that means you will have to have some kind of amino acid based on this context. So this is dependent on context dependent, specifically what position you have helical region. So this is not entirely independent modularity in nature. So for example, in position -1, in place of you, if you have in place of arginine For example, we'll see if you have arginine, that means you will always have G or some kind of C position at the first position, but you will never find A or maybe a C, for example, C at the first position. So I don't know exactly which one is precluding, but this is the context dependent I'm telling. So if you have certain residues at some position, that will give you a very, limited kind of scenario to change the second finger domain, and that limits many times the positioning of this DNA binding protein at the particular site. Now, the second limitation is the lack of specificity. And let me erase it. Now, the second is lack of specificity of some zinc finger domains, and it can generate off-target cleavage, leading to undesirable mutants and chromosomal aberration. What that means is, if you remember, I asked you to actually remember G or T at the first position, or G, A, T, something like that, and third. For example, if you have one finger domain that is recognizing at the first position G or T, second position GAT, and the third position T. So that means this will give you a lot of sequences of these. You can have one sequence, for example, GGG. You can have another sequence, for example, T, G, G. Second sequence, you can have G, A, G. So there are a lot of sequence that one specific zinc finger nucleases or domains will be able to find. So if this is not specific, that means if this is not only binding to GGG, it can also bind to GTT in TGG. It can also bind to GAG. That means it can bind to specific other sites also to create off-target effects. That means not only this site will be, mutated if you want to cleave at this site. Not only this site will be cleaved, some other positions will also be cleaved by this kind of, and that actually limits the use of zinc finger domains, because it can also recognize some off-target sequences. Is this clear? And the third main thing is, though in theory we say that we can do it, and then there are a lot of publication also, but the practicality is quite cumbersome. For example, one has to create this hybrid enzyme or different kind of nucleases linking together, and the whole process actually is very cumbersome, and the end result of very specific DNA binding sequences you will get only after many rounds of phage selection. For example, you have used the phage genome and you are selecting based on the oligonucleotides. And after first, second, third, or maybe after 10th round, you are able to specifically say that this target will actually be able to, specifically select this. And that is only, phage is only based on, lot of sequences. And when it goes into in vitro, that can still give you some law of target effect. So these are some of the limitations of your nucleases enzyme, specifically with zinc finger nucleases. So this brings me to my end of today's lecture, and we have seen today that how the first zinc finger nucleases were created by the same group, Chandrasekharan group. So he immediately actually described the FOC1 modular domains, for example, N and C terminus in 1991. And after that, immediately UBX nucleases, this hybrid nucleases he described in 1993, I guess. And after that, zinc finger nucleases he actually described in 1996. And after knowing the dimers of FOC1 in 1998, actually they started using different opposite and oriented this hybrid zinc finger nucleases. So this is the whole story of your zinc finger nucleases and FOC1 enzyme. So this hybrid nucleases were used by various labs. For example, today I mentioned about Carul lab just briefly, and then Porteous lab, who also used in GFP sequences. They together found this company which was used in monogenic kind of diseases, and they want to actually cure these monogenic diseases. And we also saw some of the limitation. For example, it had limited modularity, it can give off target effects, and third, process itself is very lengthy, and by the time you get it, it really takes a lot of time. So basically independent lab actually they order used to order these kind of hybrid nucleases from Sangamo company or some other biotech companies. So with this, I conclude today's lecture, and maybe this is now open for a question answer session. So you can ask if you have any questions pertaining to today's lecture. So there was one question that was asked by Aishwarya, and that was regarding silver staining. Had I answered that question, or still that is still open? So she asked about silver staining question, why silver staining was not used in Engelke experiment, right? So Engelke, when he used transcription factor and purification of transcription factor, that was 1980, and silver staining actually came into picture long before that, but basically it was used in histochemical assays for staining tissues and other things. But the first time usage in agarose gel was in 1979, and subsequently in 1980 they started using SDH gels for more sensitive pictures. So this is around the same time when Angelka actually paper was published. So I presume because of that closeness, or maybe 1 or 2 months gap, they were not able to use silver staining gels. So now coming to tomorrow's quiz. So tomorrow's quiz will be based on the entire lecture from lecture number basically 1 or 2, I can say 1 till 12. And if you see, if you are able to, grasp that what fork enzyme is, how it behaves, what are the dimer interfaces, what are the specificity, then how it was purified, then not much about features, because these are the crux of whole zinc finger nucleases as well as your FOC1 enzyme. In zinc finger nucleases, particular experiment. For example, if I give you this experiment or gel picture, will you be able to describe that what they have done in this experiment? So they have used this lambda phage, this DNA, and the UBX FN and other enzymes were used in this. So this kind of questions will come for tomorrow, and there are approximately 10 questions. There are small answer questions also where you have to just write one word. Basically, you can have sheets, white sheet paper with you, write your name and roll number, and start with the first question, like just write the answers. Don't write any question, just write important answer. And numbering is important. For example, if you do mistake in numberings, then it will be disastrous, for example. So all short answer questions, and then there are subjective questions, that means 2 marks and 4 marks. So those are very basic questions. For example, if you have understood the slides all over, you will be able to do all these questions, and there are no tricky questions as far as my understanding goes. So now, if you still have any questions pertaining to lecture 1 to lecture number 12, you can actually ask. Yes, Rajiv?
Yeah, hello, ma'am.
Yes.
Ma'am, I have a
Hello.
doubt from from lecture 8. Actually, you had asked this question. With regards to the footprinting assay of the transcription factor, when we incubate the DNA with in different combinations of the egg extract, the polymerase and the transcription factor, why is it that we get why is it that the DNA is protected in different patterns for the different combinations?
Mm-hmm.
Yeah, so what's the reason for that?
Uh, so what is the reason? You have attended all the lectures, right?
Yes, ma'am.
So I have explained this already with one kind of figure.
Mm,
So
uh,
if you remember protection assay, right? So are you talking about trypsin assays and then incubation of DNA together with proteins? Or your
uh,
question is totally different?
And in lecture 8, we had incubated the transcription factor, the egg
Mm-hmm.
extract, and the polymerase C, RNA polymerase C together to see which internal regions of the DNA are protected. And wherein we can see that it is protected in different ways.
Mm-hmm.
Um, like the patterns in which it is
Okay,
protected
now
is
I
slightly
understood.
different.
Mm-hmm.
Yeah.
Mm-hmm, mm-hmm. So they are looking for the only protection that is caused by transcription factor and not by RNA polymerase, because RNA polymerase will also bind somewhere, right? Because it is uh, so what they are doing three different kind of assays there. So one is the egg extract together with your RNA polymerase. One is the just the transcription factor, and one is just the RNA polymerase and together with the transcription factor. And in all three experiments, you will see that RNA is protected only when you have transcription factor, right? So that is the reason. I will come to that picture. Let me just a minute. You're
Yes, ma'am.
talking about this picture, right?
Yes, ma'am.
So here you are getting protection only when you have transcription factor, isn't it?
Yes, ma'am.
So okay. So so this is one where you have just the transcription factor, it gives you protection here, right? So when you have just the polymerase here, polymerase will not give you any protection in this region specifically. So they are not binding all alone to the region where transcription factors are binding. So that means the interaction. So this is what I'm saying, that polymerase will give you some kind of RNA synthesis, but that is not very specific. So this the role of transcription factor, if you have studied, the transcription factor will form the binding with your DNA, and then it this is the one factor which brings your RNA polymerase to this DNA for the specific start of your RNA synthesis. So basically protein-protein interaction will happen between transcription factor RNA polymerase, and there is a direct protein DNA interaction when you have these kind of transcription factor. So you have protection only when you have transcription factor involved in the reaction mixture here. So specifically or here also.
Yes, ma'am. But my doubt was like in lane number 3 and lane number 5, we can observe that the protection is offered in different ways. At certain regions in lane 5, it is completely protected, but in lane 3, not at all points it is protected from 47 to 96. Uh,
Yes. Yes,
That
that is true. And this kind of variation comes when you have more specificity, when you have incorporated with your transcription factor together with polymerase, right?
okay.
So,
Okay.
and also because these experiments are done 1, 2, and 3, and since this is radioactive picture, and specificity, the time, how much you incubate in the fridge after, radioactivity, how long hours you can, so all this actually matters when you take out the picture.
Okay.
So that is the difference. But basically, if you see, the protection is in all three cases, 1, 3, and 5, this is around the same region, and the region was actually mapped.
Okay, ma'am. So it is only because of the transcription factor and not of any other factors from the egg extract or the polymerase 3. Like
Yes,
that is the
yes.
main conclusion. Okay,
Yes,
thank
yes, because they have done all the controls here. For example, they have
you. Yeah,
control where you have polymerase 3 and egg extract, which does not protect basically in this region, right? So
yeah.
you see these kind of bands. And here also they have done controls where they have incubated RNA polymerase 3, which does not give any protection around this region.
Thank you, ma'am.
Yeah, welcome. Yes, second was who asked the question? Vishal or Sam? Vishal, go ahead.
My question is from lecture number 3.
Okay, lecture number 3.
Yes, ma'am.
So,
Ma'am,
tell me.
uh, no need to go to the slide, ma'am. It will.
Okay.
Yeah. The question was, you mentioned in point number 4 somewhere that the 25 kilodalton fragment appears resistant to any further trypsin digestion, right? So suppose I just want to know the approach, ma'am. It's not relevant to the exam, I guess. But the
Okay.
point is that suppose we use trypsin for the digestion, correct?
Mm-hmm.
But now if we get the 25 kilodalton and we just and it was not further digested, so we just
Mm-hmm.
assume that this unit is responsible for the cleavage property. But what if we digested it with some other enzyme or another chemical, then we might get some let's say 13 kilodalton or something, which will be which will be specifically responsible for the cleavage property. So I just want to know the approach, how they decide that what when they should digest with what kind of things and when they should stop. Because if I were there, I would have digested it with some other you know some
Yeah,
other.
yeah. I
Yeah.
understood your question. So basically, they are doing, no, they are knowing the sequences. So these kind of digestion they do to know the sequences within a big protein. For example, I have a protein of like 100 kilodalton, and they want to actually start knowing that what is the 5 prime amino acids and then internal and other things. So basically, for 5 prime, they go for Edman reagent, right? So one by one they can know what are the sequences, because but that specific pipeline sequencing, either Sanger or Adman, actually is very time consuming. One by one
Yes.
you have to do it, and only small portions can be done, like 10 to 15 amino acids can be released. So if you have such a big protein, you can actually take either trypsin, you can take in second container this chymotrypsin, and third container some elastin or some other enzyme, right? So you can actually have one experiment prepared where you have 2 3 different kind of enzyme, and then the same protein is incubated with 3 different kind of enzyme. Now you see what kind of patterns you are getting. That means at specifically that positions, for example, if you are getting a big sequence and a small sequence, so at least you can know that this many lysines are present in this sequence, and from second experiment you can specifically know that these many hydrophobic amino acids are present, and similarly that. So this is the type of approach they take, that okay, in a bigger protein how many these kind of proteins are there. So that is how they continue. And in trypsin and then the POC enzyme, I have to re-go that and see that what approach, but they have done it with the trypsin. And then with the trypsin, they were able to figure out that this many C-terminus domain is there, and this is not further cleaved by tyrosine, no, sorry, trypsin, and this is also giving the cleavage activity. And you are correct that they can even further reduce it down, but even after a further experiment, they were able to show that this is the only 25 kilodalton is full responsible for the cleavage activity. If you go further mutate that from the C or N terminus, then it can happen that the cleavage activity is reduced further. So this is so actually in any kind of protein you also do this kind of method, like either you create N-terminal deletion or C-terminal deletion, and then you carry out the activity assay.
Okay. Thank you, ma'am.
Yeah, welcome. Sam?
Yes, ma'am. Am I audible, ma'am?
Yes, yes.
So, ma'am, this was in reference with this is in reference with lecture 6, where you actually mention about the dimerization of the C terminal, the catalytic domain. So, when you mentioned there over there, so one experiment they showed in a gel where they show that the DNA is being cut and the like with the wild type fork one, the DNA is being cut and eventually there's a linear domain, a linearized DNA towards the end. But when they use the same thing with just with the D483A and the R487A mutants, they see an open circular structure and no linear structure. So my question, so the thing is, after that very slide, it is mentioned that when the fork one binds to DNA as a monomer, the complex is inactive. So I think I feel this is quite contradictory. So how exactly are we seeing the open circular structure if that is the case?
Okay. So this is a very important question respected to your quiz also and to your understanding as well. And I suppose you were present in all the lectures because I have mentioned the same thing in one another talk where the same question arises, that there is a open circle and why there is still an open circle, because if you mutate the interface, dimer interface, this whole the activity cleavage should not happen in either of the DNA strand. That is your question, right? But still we are seeing this digestion, right? So they have not shown in their paper, but I have mentioned that in I took that picture in another slide and I presented that apart from the amino acid which has been shown in the dimer interface, uh, not only dimer interface, but we are talking about here this mm, we are talking about, yes, dimer interface where D483 and R487 was used as a dimerization. But apart from these two amino acids, there are other amino acids also. For example, isoleucine is there, then asparagine is also there. So these are also making a prominent interactions for forming your dimer composition. So if you just mutate these two residues, there's still other amino acid which are forming this dimerization. So I suppose you have missed that class, or probably if you are present, then you have not paid attention to that one. Let me actually show that again. Just a minute. Do you remember in which lecture I had shown that figure? Anyone, if you can.
Lecture 6.
No, that is lecture 6. But after that, there was one questions, and I have uh, that was, I have to see which lecture was it. 6 or 7, I don't know.
Amino acid mutation part.
Wait a minute, I will check it. Not in 7. Uh, yes, so this one, and then there is also. So when you see this dimerization pattern happen somewhere in this alpha 4 and alpha 5, right? So this is just the domain. So here you have D and this R. This is important. But apart from that, you have this glutamine, and then there are these residues which are forming your isoleucine. So I have a better picture of of forming this. Let me show you once. Figure 15. So yes. So let me. Okay. So you see this picture. So these are, let me show you. So this is what we are talking about. Uh, s- this is again the dimer interface. So here D48 483 and R487 is not shown, but apart from that, other interfaces, for example 486, then E490, these are making interfaces. Then similarly Q486 from this monomer and E490 from other monomer is also taking part in the interfaces. Then apart from that, you have this isoleucine at 499 and this isoleucine at 450, 4538 position from both the monomer, which are also taking place for the interfaces. So this is not only these two dimers, and this was found after that paper. So this is found in subsequent actually articles. So that is why you still see a less activity, but the activity is not totally gone, and that is why you feel that this is a drastic or different what you say different sentences. Is that clear?
Yes, ma'am. So probably
Yeah.
if maybe if the other domains or if the other residues were mutated, they might have less, or probably
Yes,
these two
it
were
will.
the more prominent
Yes,
ones.
yes, yes. So if you also mutate these residues, so you will not form any dimer anymore, right? So if you mutate along with aspartic acid, if you also mutate the E490, then I feel this whole dimer will be collapsed, and then you will not get the activity.
Okay, ma'am. Ma'am,
Yeah.
I had another question, but
Yes.
this is regarding last class, lecture 11. Towards the end, you mentioned about DNA periodicity and its relationship with the linkers and the linker size. Ma'am, could you possibly explain it once again, ma'am? I'm not very sure exactly how that correlates.
Today's or yesterday's
Ma'am,
class?
yeah, maybe last
Yeah.
class, ma'am, not this class.
Okay.
Last class, you mentioned about glycine,
Okay.
serine, glycine residues being added.
Okay. So basically that is to increase the, increase the flexibility. So yeah, this was in, this I've shown in lecture number 11 itself. So this is from lecture number 11 where I showed these important residues and that are taking place for the dimer. And now coming to the periodicity. Um,
It's towards the
for
end,
this
ma'am.
one, so this one, right?
Yes, ma'am. Yes.
This or this. Okay, and this or this. So what we are talking here about, to increase the strength and specificity, one has to increase the number of zinc finger domains. For example, if you use just one domain here, it will cause the 3 base pair binding prominently from this one, right? Similarly, if you increase, it will again bind to 6 and then 9. So this is done until you have one helical segment or helical periodicity. But the periodicity of this zinc finger and the periodicity of your DNA in a helical form, because this is also turned, so they do not match each other. And to actually, overcome this kind of problem, they induced longer, linker sequences. So here they have added an artificial linkers where they have incubated like glycine, and then serine. So this kind of linker they have used number of times, for example 3 to 4 to 5 times. So that means either they are adding 15 amino acid linker or maybe 20 amino acid linker to increase the flexibility of, or to match the periodicity of this finger domain as well as with the DNA periodicity. And that is what I meant to say here with the linker. Similarly here, there are two strategy. Either you allow the longer linker between the two finger domains, which are like used 3 times, or you use 2 linker domains. So here the shorter linker domain, and then you add 2 this new finger domains here, 3 times 2 finger domains with having 2 times linker domains here. So these kind of strategy were used by a lot of labs to, and basically this was more prominent compared to if you say the previous one. So compared to this one, this strategy was more prominent because that gave better results in their experiments.
Okay, ma'am. Thank you, ma'am.
Okay, welcome. Any other questions? Yes, Surbhi.
Hello, ma'am.
Hello.
Ma'am, I have a doubt in the lecture 9, I think, where they have described the domains of the transcription factor. Like there is a 40 kilodalton which further splits. In that case, when we are like arranging the, like, ma'am, can you show that figure? Then I can ask.
Sure. Lecture number 9. We suppose this one. This. Which one? Surbhi?
Oh, sorry, ma'am. Ma'am, the previous one, the SDS page gel image.
This one?
No, ma'am, the gel image. Yes, ma'am, this one.
Okay. So what is the question here?
Yes, ma'am. So in lane 4 for the second gel, we are getting like 2 bands for this 20 kd fragment. But in this image, like you said, we will only consider the CB3 fragment, not the upper one.
No, not like that. So if you say that now basically what they did, they digested it with cyanogen bromide, and now they want to map it. So if you have this kind of coming out 20 kd fragmented into 2 pieces, and if you want to actually make the map, then how will you map it? So you can map it something like this. This is the 20 kd, for example, this one, and then the third fragment what you are getting is this one fragment. So CB3, cyanogen bromide 3, is giving you around 10 kilodalton fragment, and the rest is the longer fragment you get. So this is all the fragment from here to here, something like that. So this is the fragment. So for example, this is cut here, and this is around 10 kb fragment, and this is the other fragments. So this is the longer fragment, and this is the full protein. That means it still needs to be digested. So is this clear, or you have further question on based on this?
Yeah, ma'am. So I'm not getting, like, if 20 kd is fragmented and one fragment is of 10 kd, so another one should be of like similar length. Then why the 2 fragments are separate?
Yeah, good question. So maybe the how big is CB, 9 or 11, something like that, no?
CB3 was of 9 kilodaltons and 2 was of 11
11 kilodalton.
kilodaltons.
Yes.
Yes, ma'am.
This was 11 and this was 9. Yes. So yeah, I have not thought about that actually. So I believe that still after 9 you are still left with 11 only, right? So I will have to check this, and I know for sure that these fragments which they are telling at exactly at 20 kd, this 30 and this 40 were later actually figured out by Miller experiment, that this is exactly at 39 kd, this is around 33, and this is around 23 kd. So this was further improvement that I did not tell in the lecture because it will again cause a lot of thing. But if you read the Miller paper, there is a segment there where they have described that they have recalculated their positions, and it was figured out this was 23 kilodalton, and that is one region, and maybe some other oxidation state that we are not aware of. That is mostly probable region also, why it is running at a higher segment. So this is, if you have one as a 19 and another as 23, then this is little running at a higher position.
Okay, ma'am. Thank you.
Yeah. Welcome. Aishwarya, yes, go ahead.
Ma'am, regarding that only, like, is some dimerization or aggregation happening there?
Dimerization. So if there is a timer for this one, that means it will go for completely double the position, isn't it? One has to look into it, but I'm not sure about that at the moment.
Okay, ma'am.
Because this actually doesn't seem like, because then the if the dimer is prominent, then it should also show in other fragments also. But yeah. But it has to do something with the this what you say, the oxidation state, because that was the major cause for different size pattern of running, because it's specifically from the purification time. If you see the purification history of transcription factor 3a, so once some scientists will isolate it at 34 kilodalton, some will isolate at 37, and some will isolate at 40. So all this was a varied number of kilodaltons which were actually observed by different number of scientists, and that all had to do with your oxidation state of different amino acids, especially lysine and cysteine residues present within this, because there are a lot of cysteine residues in this. And apart from that, the major experiment they did to prove this oxidation state was iodoacetate. And only after iodoacetate treatment, and then that further smaller fragments that revealed that all these proteins are actually similar, and they also give the same similar running pattern. So it can happen that at this running pattern they are behaving little different.
Okay, ma'am. Thank you.
You're welcome. Okay, if you have no further questions, Surbi, you still have some doubts?
No, ma'am. I just forget to load the hand.
Okay. So if there are no further questions, I wish you all the best for tomorrow's quiz. Don't panic. First of all, don't panic, and don't, give like, don't think too much that, okay, what if this, what if this. So straightforward, whatever has been taught till now, only those questions will be coming. And there are no tricky questions. So if you have understood the lecture, so be at peace. Just read through the lectures and perform well in the examination. Don't write too much. Be specific in giving your answers, and specifically for 1 word and 2 marks questions, you can be very specific. Only write something where at least 4 marks questions are needed. Right there, you can actually elaborate on your answers and your concepts. That is what I want to say for tomorrow's exam. So all the best. See you tomorrow at the same time. What I will do tomorrow, I will upload the questions specifically just before 3. That means maybe 2:55. I will upload the questions on the class material side, and then I will be present here online. I request you all to be present at the same time, so that if you have any doubts, you can ask me and I can also explain what that question means. And then at the end, by 3:55, you should take the picture and upload your PDF copy in the form of roll number and send it to Gmail ID. I will also tell that Gmail ID tomorrow also. Okay. So this is what I expect from you for tomorrow's lecture. Okay. Thank you from my side. Yes, Vishal?
Ma'am, okay.
Ma'am, so total number of questions would be 10, no?
Yes, it is of 30 marks, but the percentage wise, I will include only 10% from this. Yeah, 10% from this, quiz.
No, no, ma'am. I'm
Total
asking
mark,
what the number of question, ma'am.
total number of question is 10 only, but that doesn't define anything. So be fast and be specific. That's what is main important thing.
Sure, ma'am. Thank you.
Okay, okay. All the best. Bye.
Just a question.
Yes, yes, Kohelika.
Ma'am, in the UB thorax, that is the UBX recognition site, in one slide I saw it was CC at the end, the 9 base pair recognition sequence. Yes, ma'am.
Yes.
And in the later part of the slides and the notes, it was TT. So in the slide where we are talking about the cleavage distance from the recognition site, it has a TT at the end. So I did not get that. What is the recognition sequence exactly?
The recognition sequence is T or C at the end, I believe, and that is why this question is arising, right? So I showed that, which was in which lecture was it? Let me.
Ma'am, lecture 7, I guess. Lecture 7.
Let me. So here also I showed. No, this is. So this is the UBX domain, and then this one, right? This is the CC, and the CC should be there at the end. So TT is at the 5' site.
Yes, ma'am. Ma'am, in place of CC, later on in the slides, it was TT. In the slide where we talk about
No.
the...
You have to check actually in the 5' codon. There is the codon region and the non-codoning region. You should check that. Okay. Because that will create a mistake if you are putting it. Pay attention that which sequence they have taken. They have taken both, the 5' to 3' and then 3' to 5'. And on
yes,
both they
ma'am.
wanted to
Yes, ma'am.
check the conjugation. So I
Yes,
believe
ma'am.
that. So here, this question you are right?
Yes, ma'am. As we can see in the left hand side, we have TT at the 3' end. So I got confused.
Okay. So this is not to confuse. So basically, because since see for this, they have also different kind of sequences. For example, when they used this box, right? They generated by these kind of incorporating 7 nucleic acid inside that. This
Yes,
9
ma'am.
basically. So they have used this sequence here. So there is some kind of degeneracy in kind of recognition sequence. So there is either CC, and later they used in this paper they have used a TT at the end. This is, but don't make any what you say. Don't confuse yourself. So If this is used as a sequence, so basically you can actually believe this is the sequence.
Okay, ma'am. Thank you.
So basically at the end of the sequence, so I also know that 2-3 other papers, they have different sites, different recognition sites. So but basically the 7 base pair are constant, T, T, A or T in between or GG. But at the
Yes,
end,
ma'am.
basically either they have CC or TT. So here they have taken as a TT for their experimental purpose, and based on that they have actually turned out in footprinting assay. Okay, thanks for saying this, because I wanted to actually say it, but it skipped from my mind. So if you rightly said here, so in this case you have CC here exactly, and here you
Yes,
have,
ma'am.
okay, fine.
Yes.
Okay, any other question? Okay, then I close this lecture for today's. Okay. Thank you. Bye bye.